Once again, welcome back, beta. We are going to start our new chapter, that is microbial cell culture and its application. Regarding this cultural part or the application of plants, animals, and the third is use of microbes, that is the microbial cell culture. Now the question is that what is the meaning of microbial culture? Previously we have completed plant cell culture, that is plant tissue cell culture, then animal, and now the turn is the microbial. The same thing is happening here also. As we are culturing the plant cells or the tissues or any explant to increase or to enhance the number of the cells in aseptic condition or in vitro, that is in the laboratory. So it is the artificial way of culturing the cells, either bacteria, plant or animal cell. So we were discussing the microbial culture. So now the question is that these microbes, what are the various kind of microbes included here? Bacteria, fungi, blue green algae. This is the mostly used microorganism which are used in culturing process, and they are done in the laboratory. What is the importance? Why there is need of microbial culture? We can directly use the microbes, but it is not the basic reason behind the culture project or the cultural aspect of these microbes are we have to increase their number in multiple times so that we can obtain a good quality of product, or we can obtain a good number of product in a proper guidance or caring, and that is done in the laboratory. So this microbial culture is also having, they also require some basic things. We cannot, but you can say, if I am saying the example of curd formation, that we can do in the home also, cheese production, but it is not done in the large production. The basic principle behind is production of large scale. We have to produce as large scale, that is industrial production, and then application. Various applications of these microbes that we have to study. Apart from that, here we are using the basic principles. What are the basic principles included, or what are the basic principles needed to culture the microbes? And these microbes or these microbial culture also require temperature, nutrient medium, raw material. Actually, medium or suitable pH or temperature is not able to culture the microbes. There is a couple of things required. There is a required number, and when all these number of things are in appropriate medium, in appropriate condition, then we obtain the suitable product, or that we call the desired products, what we need. So this is the requirement. Now why we are using now microbes? What is the basic concept behind to using the microbes for culturing purpose? You must know that the smaller the cell, the rate of metabolism will be very faster, and metabolism is what all the biochemical reactions that take place in the cells is known as metabolism. So these microorganisms have infinite power, or they have a very good power of the metabolic reaction. Now these culturing purpose is able to multifold, that enhance the metabolism process. So this hidden metabolic rate or metabolism is converted into the production of products. Production of products when it is under the appropriate pH, temperature, nutrient medium and the raw material. So I would like to correlate that single cell protein. One of the example is single cell protein. These single cell proteins are what? These are the proteins which are derived from the single celled organism like yeast, like some bacteria. So these single cell protein require some raw material. Apart from that, they require nutrient medium, then pH, etc. And they are able to produce the single cell protein, and the single cell protein which we are taking from the yeast. I am giving the example yeast. Okay, so this yeast is the material and that is converted into the single cell protein with the help of the nutrient medium, with the help of appropriate pH, temperature, etc. and it is being done in the laboratory. So this is the aspect behind this chapter. Apart from that, these microbial culture have increased vigorously because of the advent of recombinant DNA technology, biotechnology. Actually, recombinant DNA technology that we call a genetic engineering and biotech. So, with the combined effort of RDT, that is recombinant DNA technology, and biotech, we are able to produce the recombinant molecules. That is genetically produced molecule, and that genetically produced molecules are, for example, insulin. You know the humulin, that is the insulin is the first produced by the outcome of genetic engineering. Apart from that, therapeutic proteins. These therapeutic proteins, for example, clotting factors. Then hemophilia, this is responsible for the hemophilia. Then growth hormones, human growth hormones. Then interferons. Now, what is the meaning of interferons? Interferons are the proteinaceous chemical substance, and they also required during the immunity system, or whenever any virus is trying to enter your body system, these interferon provoke, that is they activate it and they fight with a particular virus. So interferons are the also like antibodies, and they are the proteinaceous substance. They activate upon the viral infection or viral attack. So this is the intro chapter of this chapter. Thanks for watching.